Welcome, everybody. I'm Professor Claudia Monaco from the University of Oxford. I'm Professor of Cardiovascular Immunology. And we are here at Vascular Discovery 2022 with some of the speakers of the session on therapeutic targets in atherosclerosis. So we're going to give you a small summary of our session, which has been quite exciting. I'll speak a little bit about my research and my talk. And the immune system is very heterogeneous within atherosclerotic lesions. And what we learned that we often have a view that inflammation is in general quite detrimental to atherosclerosis, and this is certainly coming out as a proof of principle. But we know that in proof of principle clinical studies, but we also know that the immune system has several checks and balances, and some of these populations are actually quite protective. We, for example, discovered a subset of resident macrophages that are quite atheroprotective. I was very excited today to share our views about how they go about and protect the vessel wall during atherogenesis. And now we're going to just go clockwise, and I'll introduce some of my co-speakers today. And Dr. Wang, maybe you want to tell us more about your presentation today.
I'm Xiao Wang. I'm a research assistant professor at University of Pennsylvania. Today, I'm going to talk about using a novel epigenome editing tools to regulate PCSK9. As we all know, PCSK9 is a therapeutic target to reduce the risk of CHD. So here, we are exploring a novel epigenome editing tools to regulate PCSK9. So in principle, it's safer compared to the traditional CRISPR-Cas9 or base editor because you're not introducing any changes in the genome.
Thank you, Dr. Wang. Now, Dr. Tufanli, do you want to introduce us on your subject?
Sure, thank you. I'm working as a postdoctoral fellow in NYU Grossman School of Medicine in the Dr. Fisher lab. In our lab is focused on the atherosclerosis regression and inflammation resolution in the mice models to mimic the lipid-lowering therapies in the humans. In my project, specifically focus on the psychological stress effects on the atherosclerosis regression. So it was known that stress is the very important and emergent risk and the prognostic factor in the cardiovascular disease in the high-risk population. So we aim to check the role of what is the psychological stress affected these atherosclerosis resolution and lipid-lowering therapies. And for this purpose, we are using very interesting animal model, the feed stress, and merge with the cholesterol-lowering therapies. So we aim to combine these two models to work with psychological stress depression effects with cardiovascular disease. So these models give us a chance to find the mechanistic links between the psychological state and cardiovascular disease. So we can planning to in future find new mechanistic targets and new therapeutics can be developed.
Thank you. And now, Dr. Weldy.
Thanks so much. My name is Chad Weldy. I'm a cardiology fellow and postdoctoral fellow at Stanford University in the lab of Tom Quatermos. Our lab is really focused on understanding the genetic mechanisms of vascular disease and using this to be able to understand causal mechanisms and discover novel therapies. Our work is really trying to understand how do we go from understanding the causal genetic variation and how it leads to modification of epigenetic enhancer function. What we do know is that this modification of genetic variation can influence enhancer function on a cell type specific manner. But a clinical observation that's been made for more than a century is that our risk of vascular disease is very much vascular bed specific, and that there's differential risk of depending on which region of vascular bed within the body. And what we don't have an understanding of are the epigenetic enhancers, how this varies across vascular bed on a cell type specific manner. So we were able to perform single cell chromatin accessibility profiling through single cell attack sequencing at 3 different vascular beds across with using a mouse model of the ascending aorta, the carotid artery, and the descending thoracic aorta. And we're able to define that there are clear differential chromatin accessibility patterns within cell types across vascular beds, really highlighting a developmental origin as well as modifying genes that are important, known to be important in regulating disease risk. I think our work is really highlighting this understanding that we need to think of our genetic mechanisms of disease or modifying epigenetic enhancer function on not only a cell type, but vascular bed specific manner.
My name's Stefan Veizades. I'm a PhD student in Patricia Wynn's lab at the Stanford Cardiovascular Institute. The work we presented at this conference is looking at the single cell transcriptome of T cells in coronary atherosclerosis. T cells have long been implicated in atherosclerosis. However, we have yet to be able to really look at them in depth from a broad, unbiased perspective. And the work we've presented has done that. And what we've been able to find is some potential targets of cytotoxic mediators involved in the development of plaque as well as the rupture.
Thank you. This has been really an excellent session. So thank you so much for staying with us a little longer.